Bom dia a todos. Primeiramente, desculpem pelo atraso, a gente teve alguns problemas técnicos com a próxima palestrante, mas tudo está dando certo. Então, a gente segue aqui para a próxima palestra. Antes disso, também eu gostaria de parabenizar todos os biólogos que estão aqui assistindo a gente. Afinal de contas, esse é o dia do biólogo. Hoje é o dia do biólogo. I'm sorry, everyone, for the delay. We had some technical issues, but everything should be working fine now. So I would like to introduce you to Dr. Cécile Albert. She's a scientist at the Mediterranean Institute of Marine and Terrestrial Biodiversity and Ecology from the Aix-Marseille Université in France. She works on the interface between functional ecology, landscape ecology, biogeography, and modeling to understand the impact of climate change and land use on biodiversity and ecosystem services. Dr. Albert, whenever you feel ready, and also Denise, which will be the one passing the slides, whenever you feel ready, you can just turn on your cameras and we should start the presentation.
Okay. Hi. Hi, everybody. Thanks for the introduction and thanks a lot for inviting me to this workshop. Denise explicitly asked me to talk today about intraspecific trait variability, which is not a lot what I'm doing right now, but which is the subject I've mainly been working about during several years, including my PhD years. And so I'm super happy to talk to you about that today. And so I entitled the presentation on the relative importance of intra versus interspecific variability in community ecology. So next slide. So as you know, the idea of functional ecology is based on the principle that we need to describe complex, diverse, and variable systems. And to do so, it's probably more efficient to use biological characteristics and similarities among organisms than purely taxonomic descriptions. And the main context behind that, Denise, I'm not sure we are really seeing the slides. Not yet. Okay, great. That's the one. The main concept behind that are the functional traits that are any measurable features at the individual level that affect the fitness of this individual. So for instance, here you can see the small plant, and we can measure, for instance, its height, vegetative or floral height, which is related somehow to its fitness. Next. And this trait is related to many other different traits in the plant. It's also related to the genotype of the individual. And so it's linked through that, through trade-offs. It's also related to many abiotic factors and to biotic interactions. And that's what makes it a complex and interesting concept. Next slide. So the idea in using functional traits, what is also very that it can be used to scale across levels, and if it's measured at the individual level or sometimes at the organ level, it also reflects what's going on at the gene level, and it can be also then averaged or aggregated at the population and at the community level to understand what's going on at these levels, and also to understand functioning at the ecosystem level. So this is interesting to go through scales and to understand what's going on from the gene to the ecosystem. And it has been used so far for many different uses. For instance, to understand or predict what's going on with vegetation. Next slide. For instance, it has been used to understand plant adaptation worldwide. So this is an old paper, but I still found it very interesting, because here you can see what's going on with several threats that have been measured on many different plants worldwide. Next slide. And then it shows that you can segregate the different species across different axes based on their strategies in terms of resource acquisition. On the one hand, you have the more acquisitive plants, and on the other hand, you have the more conservative plants regarding resource acquisition. And this can help you make functional groups and understand trade-offs among threats and among functions in the different plants. Next slide. So then these threats, they have been stored in large databases, for instance, in the very well-known TRI databases, which now gathers many different threats for many species around the world. And threats have also been used to or known to influence different processes, for instance, regarding species interactions, community assembly, or ecosystem function. And they're also known to be related to ecosystem services and their synergies and trade-offs. But the main thing is that to do that, so next slide, instead of being looked at the individual level, all these different things have generally been looked at the species level. So considering generally average threat values at the species level, instead of really considering threats at something measured at the individual at the individual level. Next slide. And this is so this is what I just said, and this has been done because generally it was thought that intraspecific threat variability was largely smaller than interspecific threat variability, and so in a way that this variability was negligible. Next slide. Despite we know that there is some intraspecific variability in threats for a long time, and it's even the basis for evolutionary processes and for competition and so on. Next slide. So the questions that I wanted to address today with you, there are three questions. First is, how much are functional threats variable? What does that imply for community ecology, which is what you are interested in? And then when do we need to account for intraspecific variability? Next slide. And then next again. So first, how much are functional threats variable? So during my PhD, we have been interested in quantifying this variability in 16 different species, all common species in the French Alps, with different life forms. So you can see here, there are some herbs, there are some grasses, some forbs, and some shrubs, and some trees. And we have measured threats over these species along within a valley, which is represented here, which is in the central French Alps. And we have measured 5 different threats: vegetative height, leaf dry matter content, specific leaf area, leaf nitrogen content, and leaf carbon content, according to a stratified and hierarchical sampling. So we organized the valley according to 2 different gradients: gradient of solar radiation and gradient of temperature. And then we stratified these in order to pick different populations of the 16 different species to reflect the variability of these threats along these gradients. Next slide. And so we found that there is a high variability of threat values along gradients. And here is only one example of threat for the leaf dry matter content. So you have on the x-axis the different species, and on the y-axis the leaf dry matter content, which is reflected for each species by a small violin plot. And you can see that there is a distribution of threat values with many values that are close to the average, but also some that are much more extreme even for a given species. And what we found generally is that this variability depends on the threats and also depends on the species. You can see here that some of the species are much more variable than others. Next slide. And this variability, we also found that it was largely structured, spatially speaking. So you can see here on the right, the bars are showing the decomposition of the variance with, let's say, a third of the variance, which is due to differences among populations. So the ones we picked along the gradient. A small part which was due to a clustering of the individuals measured within the populations, and a large part, almost half of the variance, which is due to individual differences. So individuals that are close, that are probably genetically related, and that are still very different in the trait they are showing. So it suggests that there is some plasticity in the system. Next slide. So we have tried then to explain the part of the variance due to differences among populations by the gradients we use to structure the sampling. And what we found is that there is only a small part, so here pictured in pink, of this interpopulation variance that we could explain by the different gradients of radiation and temperature. And what was striking was also that the relationship between the traits, so for instance here always leaf dry matter content according to the different gradients, the relationship were completely different for the different species. So we found a strong idiosyncrasy of the results of the relationships of traits with gradients. Next slide. And so we thought that maybe this is because each species has its own environmental requirements and its own optimal along the gradients. And so we try to explain this part of variance with the niche of the species, and it appeared to be quite promising, though it was still not explaining all this variability. Next slide. So now, because what we were interested in at the beginning was to understand how much traits are variable within species in comparison to between species, we also looked at what was going on if we are looking at all the traits together, so in a multi-thread approach. So here you can see the 5 different traits that were measured. They can be organized along 2 different axes. The first axis, which represents a large part of the variance, 50%, and the second axis, which is representing 30%. And then in this space, you can put the different individuals and species that have been measured. So next slide. For instance, here we see for Festuca paniculata, the average of the species in the multi-thread space. And each segment is representing the link between the average of the species and the individuals. So here you see all the gray stuff or representing all the individuals. And then if we put all the species together, the 16 species, next slide, you can see that you have all these species that are organized in the space, the thread space. They are kind of aggregated regarding to their life form, but also there is still some overlap between the different species. Next slide. So here we see, for instance, that we have the forbs that are on the same part of the graph, and the grasses that are together, and the legumes and the shrubs. And also we see that we are finding back this relationship among traits that leads to some species that are more acquisitive and some that are more conservative, as the one that I show you in the paper from Diaz that was at the global scale. So even in the valley, we are finding back some of these same patterns, but still, next slide, we see that there is a large part of this overall data set, which is due to differences among, within species, with almost 30% of the variance, which is due to intraspecific variability, and still some overlap among the different species. Next slide. So in the end, to answer this first question, how much are functional traits variable? So I show you some results that are from my PhD, but now since then there have been a lot of papers published on this topic and a very flourishing literature dealing with intraspecific variability in the recent years. And this literature has largely confirmed that there are strong discrepancies between threats and between species and between organisms regarding this variability. For instance, some papers have shown that there is a very high intraspecific threat variability in lichens or in tadpoles. There are also some strong discrepancies among threats, with some threats that are very variable and some others that are less variable. And there are also some strong discrepancies in the relationship between threats and climate relationships. There have also been a lot of papers showing or trying to decompose and compare the intraspecific variability and the interspecific variability. And on average, the general range for intraspecific variability is between 20 and 40% of the variance. There has been also some global meta-analysis with more than 600 different communities looking at this decomposition of variance. And if we are more or less generally always going to this range, 20 to 40%, then it also completely depends on the sampling design. So the next question is, what does that imply for community ecology? Next slide. But what I wanted to show just very quickly before that is this question of the sampling, because I think if you're interested in community ecology, you really need to think about this sampling question a lot. And so here, just small things about this sampling question. So here you can see the same graph as we saw before, with the leaf dry matter content for the 16 different species we measured, and for each violin plot in gray, which is showing the range of values that we observed for this threat for the different species. And the blue lines, they are showing the range of threat values that were obtained for the same 16 different species, but from a database, which is the Alpine database resource for species and ecosystem features. And so the plain lines, they are showing the main range, and then the extreme values that we found on the database are represented by the dotted lines. And so we see that we found in the field some threat values that were kind of more extreme than the ones from the database. So the main message here is probably that if you're really interested in intraspecific threat variability, you need to be really sure that the database that you use are really done for that and are really appropriate. For instance, it is now known that in many databases, for instance, there are lots of data, but probably not always very suitable to understand intraspecific variability, simply because data have not been collected for that in the first place. And then, yeah, it also relates to the question of standardized protocols, because at least in plant ecology, there has been a lot of work around the development of standardized protocols to measure threats. So you need to pick individuals that are in this way, extra. But the thing is that in this case, if you're always taking only adult plants that are well developed and so on, you're not really accounting for all the variability which is found in the community, and the smaller plants and the younger plants that are also belonging to the community and that are also having a role in this community. And so many papers that are dealing with intraspecific threat variability advocate that probably the protocols should be relaxed in some ways in order to better describe this variability and better understand the processes that are going on within communities. And regarding the sampling design, the sampling strategy, so next slide, there is also this question of how you need to sample communities along gradients if you want to, for instance, if you want to compare intra and interspecific variability. So here was this simulation work I did a few years ago. So on the x-axis, you have the range of light availability, which is sampled in the subset of communities that were picked. And on the y-axis, you have the percentage of variance, which is found among species, so interspecific variability. And what is interesting is that this percentage of variance among species changes, of course, based on intraspecific variability, which is represented by the different lines, plain lines, and dotted lines. But it's also largely dependent on the way you are picking the different communities. So for instance, if you pick more communities going to the light gray to the black gray, you are having more interspecific variability because you're picking many more communities along the gradients, and then you have more turnover. But also if you are picking, for instance, looking at the labels A, B, C, D, if you are picking few communities at the extreme of the gradient, or many communities along the gradient, but well organized with several steps in the gradients, you don't find at all the same decomposition in the variance. So these results need to be always linked to the way the sampling has been done, because it matters completely. So next slide. So now what does that mean? What does that imply for community ecology? Next slide. So it implies many different things, and I try to organize that progressively. So first, for instance, intraspecific trait variability changes how we compare communities. So here, if we have this community, which can be described by the different trait values and the abundances of their species, we can describe the community with its functional diversity, which will be the range, kind, and relative abundance of functional attributes. Next slide. And we can measure on that different components and metrics of this variability. For instance, the community weighted mean, which will be basically an average trait weighted by the relative abundance of the species, or the richness in the community, which would be the range of trait values obtained in the community, or the evenness, which would be how well the abundances are distributed along the trait gradients, or the divergence, which is a kind of variance, weighted variance in the trait values. And so these different metrics, then what do you do when you're wanting to integrate intraspecific variability? Next slide. So if we want to describe that and compare different communities, usually we are doing it this way with a very simple trait value and abundance as I showed earlier. But you can also do that by describing the distribution of trait for each species, what you see at the bottom. So each species has a distribution of trait, and then the distributions are overlapping. And then it means that if you compare the first row and the second row, that you're including this intraspecific variability, but only the red part on the left, so only the within population intraspecific variability. Next slide. Now, if you compare two different communities, you will have to account for the turnover, so maybe not exactly the same species in the two communities, and changes in the abundances, the relative abundances of the different species in the different communities. Next slide. But you can also account for a part of intraspecific variability, which would be that the species may shift their trait values because the conditions are different. So for instance, here, the purple bar is like going upward and the yellow bar is going downward, etc. And so here you're looking at the between population intraspecific variability. And next slide. Now you can also combine these two different parts of the intraspecific variability, and it means that you're changing your way of describing your community from what we had at the beginning, which were only single bars for each species in the community. And now in the end, you have the distribution of trait values, and you have also shifts in the mean trait values for the different species. So you're accounting in this way for all the intraspecific trait variability. Next slide. But this means that the concept and the methods that are behind are different. And there has been a lot of papers published on this issue of how to integrate these different components of intraspecific variability when you are measuring diversity and comparing communities. Next slide. So also intraspecific trait variability modifies functional diversity values. And here I show you some results from a semi-virtual experiment. So the idea is that we started from the field data I presented earlier, because we also had some data on the communities. So we had, for instance, here 46 different communities organized along the valley, within which we had 91 structuring species, for which we had real average trait values. So we have real trait values, real abundances, observed abundances, and we had also a range of coefficient of variation for the different trait values in terms of intraspecific trait variability. So we could compare what's going on if we have no intraspecific variability, so just the trait values as they are measured and averaged. And if we include, artificially include some intraspecific variability by simulating some variance around the trait value according to the different coefficient of variation that we observed in the field. And so then we made, for two different traits, vegetative height and leaf dry matter content, and 4 different functional diversity indices: community weighted mean, functional richness, raw entropy, and functional diversity. We compared what's going on if you measure these diversity indices without intraspecific variability or with simulated intraspecific variability. Next slide. So then for each community, we have the measures of functional diversity without intraspecific variability and with intraspecific variability. Next slide. And if you put all the communities together, you have a relationship like that, and you can measure an R-square. Next slide. And then we can compare the R-square for the different cases. Next slide. Meaning that low R-square means we need to include intraspecific trait variability. Next slide. So for instance, here, for the vegetative height and an increasing coefficient of variation, so we simulate intraspecific variability with increasing variance, we see that R-square is decreasing, but we are still, like for this trait and community weighted mean, we are still with R-square above 0.8. Next slide. But then if we are looking at different indices, we see that the R-square are going lower, and that there are strong differences between the indices. Some indices are more sensitive to this inclusion of intraspecific variability. And logically, the one which is less sensitive, the community weighted mean, is an average. So it's less sensitive than a variance to changes in the values. Next slide. And then we see also here, for instance, that the leaf dry matter content leads to diversity indices that are much more sensitive to the inclusion of intraspecific variability. So the indices are differently sensitive, but also the traits leads to differently sensitive indices. Next slide. And in the end, if we compare with real observed values of variability for these traits, for instance, vegetative height was variable around 0.2, 0.3 in terms of coefficient of variation, and leaf dry matter content around 0.1. And so it means that, for instance, the community weighted mean are quite relative for these indices in our communities, and they are reliable. But if you are looking at functional richness or functional diversity, then we are going to R-square that are close to 0.4, which means that the values we are measuring without accounting for intraspecific variability are probably not well representing the communities. Next slide. And so when compared with observed intraspecific values, some indices seem to be poorly reliable. Next slide. Now, Intraspecific thread variability also helps us to understand how communities respond to extreme climatic events. And here is this experimental draw that has been conducted by Vincent Young in the French Alps a few years ago. The idea here was to have greenhouses in which a part of the plots were watered and a part of the plots were not watered during the summer. And he measured the community weighted mean and the evenness in the communities for 3 different threads: leaf dry matter content, specific leaf area, and leaf nitrogen content. Next slide. And he showed that here's still an example for leaf dry matter content, that the community weighted mean were dramatically increasing as a consequence of the intraspecific variability. So species are changing their threads, shifting their threads when they are watered or not watered. And it's largely due to intraspecific variability and little due to turnover in terms of relative abundance. And regarding the evenness, so this evenness remains more or less constant with or without the watering. And it's due to 2 different effects that are contradictory. The turnover leads to a smaller evenness, but the intraspecific variability leads to a higher evenness. And in the end, there is no change, no real change in the evenness. So in this experiment, what was interesting was to see that intraspecific variability was largely driving the short-term response of the community to these extreme climatic events. Next slide. And last point is about the intraspecific variability that helps us to understand species coexistence, or at least helps us like disentangle this question of species coexistence. Here we see on the left 2 different cases where we have the different distribution of thread values for the different species. So for instance, on the top, you have narrow distributions, for instance, for the red or the purple value, they are narrow, the distributions. But when you are going to the second case with the community B, they are larger, the distribution of threads, with more overlap of the different species along the thread axis. And there are 3 different hypotheses that are suggested to understand the relationship between species richness and intraspecific variability. So either the classical theory, which would be niche theory, we should expect that there is smaller intraspecific variability when we have more species. So it's due to increased niche packing. Yeah, the first relationship on the top. You increase intraspecific variability when you have more space and less species, and so there is globally always the same overlap, so little overlap anywhere on the gradient. And this has been shown by different papers in different systems that have shown that they found this relationship, like increased niche packing and smaller intraspecific variability, narrower distribution of thread values when there are more species. There is also on the bottom this idea that what drives the system is more the individual variability. And so there is, in the end, more intraspecific variability when there are more species, because like that, the species can have individuals that are less in competition. So this has also been shown in different systems and different publications that support this hypothesis. And there is also another intermediate hypothesis, which would be neutral theory, which would say that anyway, intraspecific variability should not be directly related to the richness in the community. And there are also some papers like the one by Kupler published this year, which are showing that the relationships are more or less inconsistent across the threads. For some threads, you find something more related to niche theory, and for others, something more related to individual variation. So in the end, it's not so clear, even if we expect that this should change based on the species richness, and this should be directly related to species coexistence. Next slide. And so probably this could depend on the threats. Turcotte et al. In 2017 have proposed that coexistence should be enhanced when intraspecific variability is smaller in stabilizing niche differences than the intraspecific trait variability in fitness differences. And so that's something that we have tried to explore in a recent paper by Mainara Javier Jordani. And she did some measurements on the tadpoles. And what we found was that generally there could be something like more stable coexistence when you have, as Turcotte suggested, more intraspecific variability in stabilizing niche differences. So it's a contrast to what I wrote, sorry. Than in average fitness differences. So if we try to explain that, it would be that on the bottom right, on the bottom right corner, you have so little average fitness differences among the species. You have a strong overlap in the fitness difference, in the fitness for each species, which means that you have, for instance, good and bad swimmers in each species. They can, you have some individuals that escape well predators in all species, but the different species, they use different parts of the water column, and they can have different microhabitats or live on different food resources. So they are not completely directly in competitions. If you are going then the top right, you are going more towards competitive exclusion, because suddenly you have one species which is much more efficient in the swimming ability than the other one, but still they are not using the same resources. But you have an exclusion because they have too strong differences in terms of average fitness differences. And if you're going to the top left, bottom left, sorry, corner, then you are going towards limiting similarity problems with some species that are much more different regarding all their characteristics, and then they have more problem to be coexisting. So I think it's slide, please, to relate very seriously to one or the other, stabilizing niche differences and average fitness differences. But at least it seems like a promising tool and way of thinking in order to better understand this relationship between intraspecific trait variability and species coexistence. Next slide, please. Yeah, next again. So in the end, what does that imply for community ecology? We can see that accounting for intraspecific variability may require using new methods and new concepts, and also sampling strategies that are adapted to the question. But it may also lead to different outcomes, measures of diversity that are different, understanding of processes that is different, though it does not always help to understand or predict what is observed, because in the end, it's not always easy to say that one trait is related to one specific dimension of the fitness or of the niche. And so it leads to our third question, which is, when do we need to account for intraspecific trait variability? Next slide, please. Next slide. So, of course, we have tried to summarize that a few years ago in a decision tree. And of course, you have at the beginning some trivial cases in which, of course, you have to include intraspecific variability if you're looking in the long term or in eco-evolutionary studies or this kind of thing. Next slide. But then if you're not sure, probably you can look at what scale you're interested in, and more specifically than the spatial scale, what heterogeneity you want to include. If you're looking at something very local with very low environmental heterogeneity, maybe there will be not so much intraspecific variability. But if you're looking at something more regional or a changing world or along gradients, then it's probably more difficult to avoid measuring intraspecific trait variability. Next slide. And it depends on how much turnover there is versus how much intraspecific variability there is. So next slide. For instance, there are lots of studies dealing with that, and there are some showing that turnover is much more important, and in the end, intraspecific variability does not matter much. For instance, if you're looking at gradients of soil conditions, but in some cases, it has also been shown with gradients of land use and management that intraspecific trait variability cannot be overlooked, or when you're looking at communities over multiple gradients or over altitude. But some studies also shown that along short gradients you can avoid looking at intraspecific variability because it was not so much important. Next slide. And then if you're looking at, if you're investigating the local scale, it probably also depends on the question and the variable of interest. And next slide. So either you're looking at effect traits, so the traits that determine the effects of the plants or the organism on the ecosystem functioning. For instance, in plants, it has been shown that with the mass ratio hypothesis that ecosystem functioning at a time t is chiefly determined by trait values of the most abundant species. And in this case, maybe it's not so important to account for intraspecific trait variability. Maybe also what you're looking at is predictable from community weighted mean and has low sensitivity to intraspecific trait variability, like the community weighted mean value, because they are something like an average. And in the end, if you add just more variance to around your average, maybe the average value doesn't change so much. For instance, it has been shown also on litter decomposition that yes, there is some intraspecific variability, but in the end, it doesn't help to understand the variability in litter decomposition, despite it's interesting to knowing there is some variability. And on the other side, so next slide, please. You can also look at response traits, for instance, traits that are associated with the response of plants to environmental factors. This is the case when looking at community assembly processes in which distribution of trait values are of interest, or when you're looking at measures of diversity that are more sensitive to the inclusion of intraspecific variability, like functional diversity measures, because there are variances. And so if you add some variance in your distribution, then the variance measures will change and will be different. And also, if it's related to filtering and niche differentiation, because habitat intraspecific trait variability is largely related to that and to how species respond to different things like disturbances or stresses. And also, if you're looking at coexistence, because there are so many things I think we have not yet explored regarding this question. So in these cases, probably ITV cannot be neglected, but I think all this is still going on, even if I'm working less on these questions right now. There is still many papers being published on this question and showing that, yeah, in some cases, it really matters to account for ITV, and in some others, much less. And next slide, please. So as a conclusion, intraspecific trait variability is not, or at least not always negligible. At least it is quite important. There is some variability for sure. And it's not only a question of quantity, because even if it's generally smaller than interspecific differences, it doesn't mean that it doesn't change what we're measuring. I showed it, for instance, for the functional diversity measures. You can have very different functional diversity measures, even for a small amount of intraspecific variability, even if it's 10% or 15%, sometimes it can really change your measures of functional diversity. And even in this paper, we went further and we showed that it can completely change your relationships that you're measuring when you're looking, for instance, at relationships between functional diversity measures and gradients. So maybe you can have a positive relationship if you don't account for intraspecific variability, and a negative one if you're looking at intraspecific variability. So I think it matters to really look at that and understand in which circumstances we can omit or overlook intraspecific variability, and in which circumstances we cannot. And simulation studies together with field measurements can be a good help, and yeah, I think there is some potential still to better understand that. Next slide. Thanks a lot for your attention.
Thank you, Dr. Albert. It was a great talk, and then I'm really convinced that interspecific variability is important. We do have a few questions for you. First one is from Mayara Vescovi-Assis. Hi, Cécile. Thanks for the nice talk. What is your opinion about considering trait covariation as an aspect of ITV? What is the importance of covariation to bring new insights about community processes?
Yeah, I think there is great potential. I think it's really important to look at several threats at the same time. Maybe I didn't say it so much, but because for several reasons, because they are not, like, it's impossible to relate one threat to one process or one dimension, like I said, niche or fitness, but they are all together related to that. And so looking at one threat individually is probably less interesting. And also in many cases, it has been shown that you can have one result for one thread and another one for another thread. So just looking at one thread is generally not very efficient, but looking at all the threads together, I think is much more efficient. And there are already some papers looking at that.
Okay, next question is from Pamela Friedman. Thank you very much for your talk, Dr. Albert. Do you think that species that present higher interspecific variation also have higher individual with niche, or not necessarily? Also, do you think it is possible that species with higher interspecific variation could attract more interaction patterns as pollinators? More interaction partners.
For the niche, at least for the results from my PhD, clearly it was not related. Yeah, we did not found any relationship between the niche width and the thread variability. And for the relationship partners, I don't know.
Yeah.
Sorry.
Okay.
That was the question, right?
Yeah, it's about pollinators. If it could attract, yeah.
Yeah, maybe there are some response about this question in the paper by Küppler 2020. At least they look at intraspecific variability for floral threads as well.
Okay. Next question is from Sebastián Felipe Sendoya. Thank you for your great talk. What do you suggest to deal with interspecific variability in functional traits that may not present a continuous variation or present a multimodal variation, such as in social insect species with castes, with an alternative reproductive strategies, or even species with strong sexual dimorphism? Do you want me to repeat it?
No, yeah, I'm not sure. I think, yeah, if you have some, if you have multimodal distribution, I think it's even more important to account for that. But yeah, maybe not in the end. If it depends if the multimodal distribution is highly picky or not. But I think all the questions I raised, or yeah, what I said was still applying here. It depends on what you're looking at in the end, and if it variability around the means is important or not for what you're looking at, I guess.
Okay. Bianca Zorger have two questions. First she says, great talk. First question is, what do you suggest in relation to interspecific variability measurement in areas with high diversity of species, like the Amazonian forest?
What do I suggest regarding what? If we should account for it, or?
Yeah, that is the question. I think she meant to account for it. So maybe if it is important.
Yeah, I think it's important. There are many papers dealing with intraspecific variability in tropical forests. The thing is that it's more complicated because it's often complicated to know the species. So it's costly to know what species you are dealing with. But we have a publication now submitted about this question of how you should sample. So I hope it should be out somewhen later this year, which is how you should sample in this system, and in order to still see the effect of intraspecific variability regarding community assembly processes. And yeah, the idea is that you still need to have some still a large part of the individual measured to see this effect, the effect of intraspecific variability and interspecific variability on assembly processes in these communities.
Okay, her next question is, what do you think about using physiological traits, hard traits, in interspecific variability, contribution in ecosystems with low morphological trait changes? Like, what do you think about using physiological traits in ecosystems with low morphological trait changes?
I don't really know. Yeah, the thing is that if the physiological hard traits are really hard to measure, then probably looking at intraspecific variability is like a nightmare. Yeah, I don't really know.
Next question is from Ramiz Ahmad. Thanks for the brilliant talk. How analyzing the variance in addition to mean can be useful in case of research synthesis?
In case of research synthesis?
Yeah.
I think it's always a good idea to look at the variance because means are often misleading. But again, it depends on the question, I guess. And yeah, I'm not completely sure what it means, research synthesis.
Yeah. Next question is from Jacqueline Zeni. Do you think human-induced changes can decrease interspecific variation in functional traits, and how this could influence in species persistence?
Sorry, could you repeat?
Do you think human-induced changes can decrease interspecific variation in functional traits, and how this could influence in species persistence?
If intraspecific variability can influence the species persistence, for sure. Yeah. But yeah, then I mean, it's open all the question about adaptation, and probably also depends on if like the time span we're looking at, if it's related to the adaptation of one individual which is already present and will adapt shift its trait values, like I showed with the extreme drought experiment, or if it's more about adaptation of the new generations to the new environmental condition. But yeah, in this case, I think it really matters.
Okay. Next question is from Veronica Zepeda. I wonder if interspecific variability may be seen as a potential of species for plasticity. And if the answer is yes, can it be used as a measure of variability as a trait?
I don't think we can use variability as a trait because, as I said earlier, at least not like a functional traits, because it should be measured at the individual level. So you cannot have a variability at this level. But is that related to species? The problem is that for now, I think most of the results that have looked at this question, they have not shown that some species, like for instance, some groups of species are more variable than others. It's always completely idiosyncratic, at least so far that I know. And so it's kind of complicated to relate variability to something in particular, I think.
Okay, next question is from Helena Galvanese. Could be high ITV change the interspecific competition to such level that it could be considered or approximated to interspecific competition?
I know that some people are working on that, but I can't tell you more about that.
Yeah, and that was so far the last question we had, but I have a question of my own. It's a more general question. So regarding conservation biology and ecosystem services, some people, I know that some people sometimes argue that we should move from conserving species to conserving functional traits. Do you have any thoughts about that? What do you think about that?
I think it should be a trade-off because what you want to keep is more functionality and ecosystems that are going well and functioning well. So I guess if you try to keep only threads and diversity of thread, but it's still always the same species, probably at one point it's an issue, and you will still lose some variability, some of the variability, or some of the potential of functioning and evolutionary dimensions, evolutionary potential. Yeah.
Okay, next question is from Diana Bertual Garcia. Thank you for the great talk. Do you think interspecific trait variability can impact, and if yes, how, can impact correlations between response and the effect and effect traits and the consequential inferences about community resilience? Let me repeat the question. Do you think interspecific trait variability can impact correlations between response and effect traits and consequential inferences about community resilience?
I'm not sure I understand well. Probably it relates to the previous question about relationships among threads and integration among threads. I think for sure regarding this, we need to look at intraspecific trait variability.
Yeah. Do you have any idea of how it can impact? I think that was part of the question.
No, not really. I think it depends on the system and.
Okay, I think that's it for now. Thank you very much for being here with us. And again, we are sorry for the technical issues we had before, but
I'm sorry.
I think
it went well. Yeah.
Yeah, it went very well. Thank you again for the great talk.
Thanks a lot. Bye.
Okay, bye. Now for the people on YouTube, we should be back at 2 p.m. with a talk by Leno Farik. And E para o pessoal que está acompanhando a gente pelo Zoom, a gente deve voltar agora às 10h30 com a palestra em conjunta do professor Tiago Gonçalves Souza e Fabrício Tereza. Muito obrigado a todos por participar.